IDBac couples mass spec data from protein and specialized metabolite regions of unknown bacterial isolates to rapidly discriminate between isolates based on both their identity and their potential environmental function.
Our open source software extends the utility of existing MALDI TOF-based microbial identification strategies. This extension includes analysis of specialized metabolism as an additional way to differentiate between colonies with similar phenotypes. A major challenge in characterizing unknown microorganisms is distinguishing closely related isolates. IDBac provides researchers an easy and fast way to characterize isolates through protein and small molecule profiling.
IDBac provides a visual comparison of specialized metabolite production within a bacterial sample and can provide insight into a broad range of research topics, from ecological studies to drug lead discovery.
There are many ways that MALDI data can be saved between and among instruments. If you have trouble getting files to work with IDBac, submit an issue to IDBac's GitHub.
Using a sterile toothpick, transfer a small portion of a bacterial colony to the appropriate spot on a clean MALDI plate. Spread the bacterial colony evenly over the spot so the spot appears as flat as possible. Overlay 1 microliter of 70% mass spectrometry-grade formic acid onto the sample and matrix control spots and allow the acid to air dry in a chemical fume hood. Next, add 1 microliter of previously prepared MALDI matrix solution to the sample and matrix media control spots and allow to air dry completely. After setting up the MALDI TOF mass spectrometer, acquire the spectra. Save the protein spectra in one folder and the specialized metabolite spectra in a second separate.